Dear students, in today's lecture, let us make an attempt to know about techniques in insect pathology. Introduction. Students, in insect pathology, it is one of the broad spectrum disciplines, includes wide array of scientific branches, namely epidemiology, etiology, symptomatology, diagnosis, dissection, bioassays, propagation, etc. Accordingly, various tools and techniques are employed to understand pathological aspects of insect species. Therefore, one has to explore all the branches in detail to get a deeper insight on diversified techniques used while studying the insect pathology. Hence, it is like an ocean, but one need not to explore the depth and breadth of an ocean. A bowl full of water is enough to study and understand the ocean and its constituents. In the present talk, such an attempt has been made and highlighted important aspects of insect pathology and few techniques commonly employed in this regard. Insect pathology is one of the branches in entomology. It has grown steadily since its inception as a formal academic discipline in the mid 1940s. Several researchers have contributed to the field of insect pathology. Lacey in 1997 has detailed techniques for the study and implementation of entomopathogens. He has also published the methods for the study of major group of entomopathogens, their discovery, diagnosis and their initial identification. Under insect pathology, microbiology, virology, mycology, nematology, biological control and integrated pest management are also included. However, in the present talk, multidisciplinary approach has been made to cover the pathological aspects, classical techniques, methodology used for the identification, isolation, propagation and preservation of bacterial pathogens. Concomitantly, the applied side of insect pathology covers the commercial development of bacterial, viral, fungal and nematode entomopathogens as microbial control agents. Their use by organic and conventional growers for the microbial control of arthropod pests of food and fiber crops, for protection of forests and health related and abatement agencies, for control of insects of medical and veterinary importances has also increased. Hence, entomopathogens are employed in all the major type of biological control and conservation movements. Thus, insect pathology not only deals with harmful aspects of pathogens, but also it covers beneficial aspects of entomopathogens. Further, the systematic survey, pathogen biology, host pathogen interactions and pathogen identification techniques are important. In this text, attempts are made to provide general guidelines for the detection of disease, identification of pathogen, handling of infected organism and initial diagnosis of diseased insects. Now, let us know about diagnosis. The entomopathogens cause diseases in insect that result from the direct effects of infection. An astronomical number of entomopathogens cause disease and the number of insect hosts in which to find them is equally enormous. However, it is a Herculean task distinguishing one disease from another based on the signs and symptoms of the disease, pathogen etiology, pathogenesis and other characteristics is the process of diagnosis. Stenios in 1963 has described the diagnosis of insect diseases as one of the most important and complex branches of pathology. The elements and background of diagnosis of insect disease are published by Stenios and Marsh in 1962 and Stenios in 1963. The diagnosis is divided into two main categories, namely the gathering of facts concerning insect disease and their analysis. However, disease and death of an insect is not always an indication of infection with entomopathogens. information on non-infectious diseases in insects due to non-microbial causes, for example, poisoning, mechanical and physical injuries, and nutritional and metabolic diseases, is also to be looked in carefully while diagnosis of infected insect. In this presentation, emphasis has been made on infectious diseases of insects that are caused by different pathogens and presents general guidelines for their identification and diagnosis. Moreover, advanced diagnostic tools such as variety of microscopic techniques as well as microbiological, biochemical and molecular techniques for the identification of specific pathogens, one has to see other text. Now, let us know about collection of diseased insects and pathogens. First, data collection. At the time of diseased insects collection, following information should be recorded: name of the insect, genus or species name of the insect if possible, stage of the insect, example adult, egg, larva or pupae, pathological conditions of the insect, behavior of the insect, location where the insect is collected, name of the host plant or habitat whereby the insect is collected, prevalence of disease, that is the severity of the disease, additional information pertaining to specific pathogen groups, example bacteria, virus, fungi, protozoa, nematodes, etc., location, elevation and climatic conditions, date and time, number of diseased insects collected, geographic region, date as well as important detail of the sampling procedures used. Proper labeling is also essential to avoid mixing up of samples, and finally the name of the collector. Let us know about diseased insects. Both living and dead insects that are infected with various pathogens are found in virtually every place, including natural, terrestrial and aquatic ecosystems, agroecosystems, in laboratory and commercial insect colonies. First, one should notice the presence of diseased insects. Thus, a visual search of habitat of interest or insect colonies in the laboratory for insects with atypical appearance is a common means of collecting diseased specimens. While many pathogens are usually present at low levels in insect field populations as enzootic diseases, they are most easily discovered during epizootics when there is an unusual abundance of diseased insects. Recognition of diseased insects in the field or subsequently in the laboratory initially relies on gross pathology and presence of pathogens. Now, examination in the laboratory. Diagnosis based on gross pathology would not reveal clearly name of the disease. Thus, it requires microscopic examination. The specific signs and symptoms help assist while identifying the different groups of entomopathogens. Moreover, color, odor, behavior or other causes such as chemical insecticides, poor food quality, etc. are also responsible for developing specific symptoms. Under such circumstances, considering the gross pathology and critical microscopic examination of the body contents of the diseased insect under good laboratory practices would help examine and identify the disease accurately. Instruments required for microscopic observation of diseased insect. A very important tool for identification of pathogen or etiological agent is light microscope and bright field microscopes. Electron microscope, transmission and scanning microscopes, forceps to handle and release apart the diseased specimens, mounting needles to aid spreading of body contents, diamond marker for frosted slides, a resistant marker to indicate any of the reagents employed in the staining procedures, watch glasses to prevent smears by decontamination of all instruments by rinsing in alcohol and wiping or flaming devices, and for microscopic observation of an insect pathogen, high quality optical instruments along with sterile standardized conditions are essential. A high quality phase contrast microscope is essential for examination of pathogens, example bacterial spores. High quality tissue culture equipments, example a wire needle to transfer a small part of one colony, an agar plate, a sterile tube containing sporulating medium, light microscope under oil immersion. Now let us know about the symptoms. Insects that are infected with entomopathogens often manifest characteristic symptoms and signs of diseases. The important symptoms are color changes, luxuriant growth of the pathogen on outer surface of the insect, signs of the pathogen or etiological agent inside the host. Those are visible through the cuticle, dysentery, peculiar behavior including lack of feeding or unusual position on host plants, tremors, mummification, fragility or hardening of the integument, noticeable differences in size, aborted molt or pupation and etc. However, in some cases symptoms of disease may be very subtle or not initially apparent. Example, sublethal effects such as parasitic castration. Some pathogens may be occult, example occult virus. Color changes. It is often one of the first symptoms of diseased insects to be noticed. This phenomenon is observed in both living and dead diseased insects. Color changes in living insects is due to entomopathogens, which are usually associated with insects that have semi-transparent to transparent integuments. Example, white grubs and larval forms of weevils, hymenopteran larvae and many groups of aquatic insects, most notably the dipterans. Blue iridescence in beetle grubs, mosquito larvae and certain lepidopteran larvae indicates an iridescent virus infection. The iridescence rapidly disappears with the death of the host. Sometimes a broad array of color changes are seen in dead insects. It is ranging from white, grey, red, orange, yellow, blue, green to brown and black. Moreover, melanized areas of the insect cuticle are often due to immune responses of the host as a result of invading nematodes, fungi, baculoviruses and microsporidia. Virus infections and several species of fungi may be observed through the cuticle of living insects also. Preparation of specimens for initial laboratory examination and or isolation of suspected pathogens. To enable detailed examination or isolation of the causative agent, the specimens will usually require further preparation. The following general procedures are commonly used for entomopathogens. First one is surface sterilization. When a non-contaminated sample of diseased tissue is needed for inoculating media or injecting into healthy insects, it requires surface sterilization. Therefore, to surface sterilize, amine and gepyrion chloride tween 80 with NaClO are used. Place insect in 70% alcohol for a few seconds to facilitate wetting the specimen, then rinse briefly in distilled water, later place in dilute sodium hypochlorite for 1 minute, then rinse briefly in 2 to 3 changes of sterile water and blot dry with sterile filter paper. However, care should be taken while surface sterilize the very small insects because it will kill the entomopathogens inside the host. In this case, the removal of spores from the tips of conidiopores is accomplished by touching the most distal spores to a minute amount of sterile media on the tip of sterile pin and then inoculating an appropriate medium in a petri dish. Now, dissection. To examine individual organs and tissues, one has to carefully dissect the diseased specimen in saline solution by using dissecting instruments. Quarter strength Ringer's solution provides a medium that is more osmotically compatible with the tissues and pathogens than water. Dissecting instruments fine tipped forceps, microscalpels, iris scissors, stainless steel minuten pins mounted on matchsticks, other precision dissection tools, slides, small camel hair brush, cotton tipped applicator stick, etc. Koch's postulate study. It should be conducted for confirmation of infections and infectious agents. It becomes the final step in the diagnosis procedure. Preparation of inoculum. Inoculum preparation for Koch's postulate tests require isolation and culture of the putative pathogen. Otherwise, inoculum is prepared by using field collected or laboratory infected hosts. In addition, some obligate parasites require production in intermediate host. There are separate procedures for isolation, cultivation, propagation and determination of the pathogenicity and virulence of particular microorganisms or nematodes. Students, now let us know about few techniques followed while diagnosing the bacterial infected insects. Bacteria. Bacteria constitute a large group of prokaryotic microorganisms. The vast majority of bacteria in the body are rendered harmless by the protective effects of the immune system and some are beneficial. However, several species of bacteria are pathogenic and cause infectious diseases in animals and also in human beings. The important infections caused by bacteria are cholera, syphilis, anthrax, leprosy and bubonic plague in human beings. Moreover, the most common fatal bacterial diseases are respiratory infections, with tuberculosis alone killing or more than 2 million people every year in the world. Bacillus species are ubiquitous, can be isolated from soil, leaves, trees, animals and dead insects. While searching for a particular species pathogenic towards a specific host, example insect, one must look for diseased host in their natural habitat. However, there are no hard and fast rules for pathogen prospecting, but thoroughness and perseverance are essential. Then collect each sample in a separate sterile screw capped tubes or specimen cups to avoid contamination. It is important to record all the information concerning sampling, especially when different people are involved in sample collection. Isolation. After arrival to the laboratory, each sample is divided into different lots and each lot is added to screw capped tubes containing 10 ml sterile water. For insect samples, place the insect into tubes containing 1 ml of sterile water per 0.2 to 0.4 gram of insect. Homogenize the sample by adding between 80 to 0.5%. Then proceed with heat treatment. For water samples, bacteria and spores can be concentrated by filtration through a 0.22 millimeter filter. The filter is then placed in a tube containing 10 ml sterile water. Briefly, vertex each tube, then proceed with heat treatment. Let us know about heat treatment and multiplication of bacteria. Heat the samples in a water bath at 80 degrees centigrade for 10 minutes and then chill tubes rapidly on ice. This step kills most vegetative cells of bacilli and non-spore forming bacteria, thereby enriching for spores of Bacillus species due to their heat resistant nature. After allowing the solid content of the tubes to settle, plate 100 ml each of the heated sample and dilutions of the heated sample onto a petri dish containing a growth medium. Incubate plates for 24 hours at 30 degrees centigrade to allow for bacterial growth. Growth medium can vary and include a sporulation medium or more typical media, example nutrient agar medium. These conditions do not allow for selection of all bacteria just for the most commonly known entomopathogenic bacilli species. Examine each plate for bacterial growth using a fine sterile loop. Transfer each colony to 10 ml growth media in sterile tubes and shake at 250 rpm on an orbital shaker for 48 hours at 30 degrees centigrade. Check microscopically for purity and stage of culture. Then plate a loop full of the suspension to isolate single pure colonies. Incubate plates at 30 degrees centigrade for 24 to 48 hours. These single colonies will be used for preparing bacterial suspensions for further cultivation. Identification and preservation and bioassay also. Factors that influence bacterial growth include physical requirements, example temperature, pH, oxygen, osmotic pressure, and nutrient requirements, example source of energy, carbon, nitrogen, minerals, water, and growth factors. For example, growth of Bacillus thuringiensis is typically done at 30 degrees centigrade. Bacillus thuringiensis strains can use a carbohydrate source for growth, whereas the other Bacillus species is unable to metabolize carbohydrates and requires a proteinaceous growth medium. Amino acids are the preferred nitrogen source for other Bacillus species, which also requires the vitamins biotin and thiamine to grow and calcium and manganese ions for sporulation. Moreover, there are many media that can be used to cultivate bacilli. This medium has provided reliable and reproducible growth, sporulation, and production of parasporal bodies in both cases. Further, identification of the major groups of bacilli species are made by using identification keys using a minimum of phenotypic characters. For simplification, a key for 22 of the most frequently encountered bacilli species is presented. Gordon et al. In 1973 arranged bacillus species into 3 morphological groups based on spore shape and swelling of the sporangium. Group 1 species produce terminal endospores that are oval and do not cause the rod-shaped bacterial cells to swell. These are divided into 2 classes. Class 1 having a rod width greater than 0.9 mm and class 2 having a rod width under 0.9 mm. The majority of the group 1 bacteria still reside within the genus Bacillus. Group 2 species produce oval endospores that swell the sporangium. Many of the species in this category have been reassigned to the family Penibacilliaceae and include bacteria of the genus Brevibacillus and Penibacillus. Group 3 species contain round spores and a swollen sporangium. They are Lysine-Bacillus spiraceus. Identification of bacilli using molecular based techniques. Traditionally, identification of unknown bacterial species has been achieved through determination of phenotypic traits. Analysis of 16S rDNA gene sequences has been a powerful tool to identify isolated bacteria. Now let us know about Rickettsia. The name Rickettsia is often used for any member of the Rickettsiales. It is a genus of non-motile gram-negative, non-spore forming, highly pleomorphic bacteria occur in 3 forms, namely cocci. It is with 0.1 micron meter in diameter, rods. It is with 1 to 4 micron meter long or thread-like 10 micron meter long forms. Rickettsia is one of closest living relatives to bacteria that were the origin of the mitochondrial organelles that exist inside most eukaryotic cells. Rickettsial organisms possess true cell walls similar to other gram-negative bacteria. Being obligate intracellular parasites, the Rickettsia survival depends on entry, growth and replication within the cytoplasm of eukaryotic host cells. Despite the similar name, Rickettsia bacteria do not cause rickets, which is a result of vitamin D deficiency. Instead, they are named after Howard Taylor Ricketts in 1871 to 1910, who studied Rocky Mountain spotted fever in Bitterroot Valley of Montana and eventually died of typhus after studying that disease in Mexico City. Rickettsia are more widespread than previously believed and are known to be associated with arthropods, leeches, and protists. In March 2010, Swedish researchers reported a case of bacterial meningitis in a woman caused by Rickettsia helvetica, previously thought to be harmless. Rickettsia species are carried by many vectors, namely chigars, ticks, fleas, and lice, in turn which cause disease in human such as typhus, rickettsial pox, Butonouse fever, African tick bite fever, Rocky Mountain spotted fever, Flinders Island spotted fever, and Queensland tick typhus or Australian tick typhus. Rickettsia species are also been associated with a range of plant diseases. Spotted fever group. Rickettsia rickettsii cause Rocky Mountain spotted fever in Western Hemisphere. Rickettsia acari cause rickettsial pox in USA and parts of Russia. Rickettsia conuri cause Butonouse fever in Mediterranean countries, Africa, Southwest Asia, and India. Rickettsia siberica cause Siberian tick typhus or North Asian tick typhus in Siberia, Mongolia, and northern China. Rickettsia australis cause Australian tick typhus in Australia. Rickettsia felis cause flea-borne spotted fever in North and South America, Southern Europe, and Australia. Rickettsia japonica cause Oriental spotted fever in Japan. Rickettsia africae cause African tick bite fever in South Africa. Cultivation. Rickettsia cannot live in artificial nutrient environments and is grown either in tissue or embryo cultures, example chicken embryos. The method of growing Rickettsia in chicken embryos was invented by Ernest William Good Pasteur and his colleagues at Vanderbilt University in the early 1930s. The classification of Rickettsia into three groups, namely spotted fever, typhus, and scrub typhus, was based on serology. This grouping has since been confirmed by DNA sequencing. All three of these contain human pathogens. Similarly, for viruses, fungi, protozoan parasites, and pathogenic nematodes, various specific techniques are followed to understand the gross pathology and pathogens involved in infections. Conclusion. Insect pathology is one of the broad spectrum disciplines require wide array of scientific techniques to understand the gross pathology and pathogens involved in the disease outbreak. Therefore, insect pathologists should be familiar with epidemiology, etiology, symptomatology, diagnosis, bioassays, propagation, cultivation, and other techniques. Further, insect pathologists should have the knowledge of microbiology, virology, mycology, nematology, and protozoology to deal with diseased insects. Moreover, insect pathologists should have the knowledge of conducting the systematic survey and diseased insects collection identification. Hence, it requires multidisciplinary approach. Thus, in the present talk, an attempt has been made to highlight certain aspects of insect pathology and few techniques commonly employed while detecting few microbes. Thank you.